Good afternoon. My name is John Kerwin. I'm the Senior Vice President of Operations at Ascidian Therapeutics, and hopefully for the next 14 minutes I'll try to convince you why we're redefining the treatment of disease by rewriting RNA. Ascidian Therapeutics, we like to claim that we're a differentiated and capital-efficient company that's really focused on three primary things right now. We're looking to advance our lead asset, ACDN01, through the clinic. Second, we're looking to advance our awesome partnership with Roche through key milestones. And finally, we're looking to advance from our preclinical pipeline additional IND-ready molecules. Through capital efficiency, where we can try to leverage existing technologies, existing clinical strategy, we can really leverage our differentiated platform rapidly into the clinic. And our differentiated platform we like to call as RNA exon editing. In RNA exon editing, we're able to replace large swaths of RNA at the kilobase scale with a single administration and with a single vector. And really the awesome thing about this is it's bifunctional. There are two things that are happening with an exon editor molecule. Not only are we able to silence a mutated form or a toxic protein, but simultaneously also able to express the wild type, and again, all in a single administration. The collaboration with Roche is fueled by several neurological targets that we partner with them with a nice upfront. And this is further validation that our exon editing technology is fueled by some of the in vitro data that I'm really happy to share with you over the next couple slides in our in vivo data. Our lead molecule, ACDN01, so this is a gene therapy exon editor for the treatment of Stargardt's disease. I know the gentleman from Ocugen shared a little bit about Stargardt's, so sorry if this is going to be repetitive. But Stargardt's being an ocular indication is a huge, huge market, and we're really excited about providing the large number of patients, not only in the US and the ex-US, with a therapeutic intervention like exon editing. What I'll share with you today is data that demonstrates that our exon editor is safe and it provides durable expression of our molecule. We have a clear clinical path. We have an open IND. We have patients are being dosed, and we have fast track designation. But more importantly, one thing that we're proud of is that this is the first RNA editing IND opened across all RNA editing modalities. We're super happy with that. We're also happy with the recognition that we've received. In 2024, we were both a Endpoints 11 as well as a Fierce Biotech 15 award winner. We're really proud of the achievements that we had, but it's the wind behind our sails, and hopefully we'll be able to convince you with what we've been able to use with that momentum. 6 key aspects of our differentiated technology. First, we're editing RNA. We're not touching the DNA. Second is we're using a single agent. In ACDN01, it's an adeno-associated or an AAV vector. And we're replacing whole exons, multiple exons at the kilobase scale. With doing this, we actually maintain native gene expression, so we're not touching the endogenous gene expression of the gene that we're trying to modulate. And we do all this without foreign enzymes. We don't use nucleases, we use endogenous machinery. And for those that are interested, I'll give a short molecular biology lesson on that in a little bit. And we do all 5 of those in vivo. All right, so the molecular biology lesson. So canonical method of protein expression. We start with DNA, then we move to pre-mRNA, mature RNA, and then the protein. So in a disease state, or if there are mutations in a DNA, that's carried over through the pre-mRNA, the mRNA, and then the protein. The key step in translation of a pre-mRNA to mRNA is a cis-splicing event. So pre-mRNA contains both introns and exons. Exons is where the protein is coded. And there's a cis-splicing event where the introns are kicked out. They're booted out, and we splice together the exons. With exon editing that we do in ACDN01, we first deliver this with an AAV molecule. With ACDN01, we do this in a subretinal injection using AAV. This exon editor contains some of the key hallmark attributes of a traditional protein expression with AAV. So we have a promoter, and that drives for the constitutive expression of our exon editor. So it's constitutively expressing out this RNA editor molecule. And using endogenous machinery, the spliceosome, it's able to recruit these donor exons and outcompete that cis-splicing event. And this is called trans-splicing. So we're transplicing in corrected forms of that pre-mRNA, and it splices together with the patient's native genes, which we're not touching. And when this translates into a full-length protein, it's the functional wild-type copy. A little bit more about Stargardt's disease in our clinical program. There's a huge unmet medical need with Stargardt's patients. We estimate at least 30,000 patients in the US. The number could be much higher. But the really tricky thing about Stargardt's is the heterogeneity of not only the number of mutations, then the location of mutations. This permits a really, really large challenge for someone applying a base editing molecule or a very precise DNA editing molecule for this. With our program, we're replacing the first 22 exons and leaving the remaining 28 exons untouched in that cis-splicing event. Again, this is delivered clinically using a pre-validated subretinal injection. Over the next slides, I'm going to describe a little bit more about Stargardt's in the in vitro and in vivo data. So first about Stargardt's, the really awesome image of an eye, which might look gnarly to some of you. The black part is a lesion, and this is a hallmark indication of a patient with Stargardt's, and this is what occurs when the photoreceptors die and there's atrophy. This is a hallmark sign of macular degeneration, is the growth of this lesion. It's in the central acuity of a patient, so this leads to really a loss of independence. The patients lose their license, inability to see loved ones in front of them, and a complete absence of that central acuity. This also provides Ascidian and ACDN01 a quantifiable and approvable endpoint. The macula itself presents not a big challenge for subretinal administration, so with a single injection we can cover the entire part of the macula with our ACDN01 and apply that exon editor to the damaged tissue. And our ACDN01 is designed to prevent photoreceptor death, so prevent the growth of that lesion, and that leads to that primary endpoint of a quantifiable and improvable endpoint. All right, on to some cool in vitro data. This is kind of the journey that started in 2023 as we advanced this molecule to the clinic. And like every other drug development company, we start with an in vitro-based study. And the figure on the left is demonstration that we can express full-length ABCA4 protein with our exon editor. And then we wanted to translate that full-length protein was functional. So for those that are familiar with Stargardt's, one of the key enzymes in there is mediated by an ATPase enzyme, and we can demonstrate that our full-length protein exhibits ATPase function. The next study was to use human retinal explants. And I'll pause for a second here, as this is a really good example when we go to the MVA or the DMV and we check donor, I'm a donor. This is a really good example where donors can partner with drug development companies to advance these awesome therapies. So from donor tissue, we were able to isolate human photoreceptors. So these are tissue relevant for our exon editor. And in vitro, what we're able to demonstrate is a measurement of RNA replacement. So these are damaged tissue, and we're measuring at least 40% RNA replacement with our exon editor in vitro. And this is over 21 days. So our awesome scientists in Brighton were culturing these human retinal explants for almost 3 weeks to demonstrate exon editing in vitro. So the excitement started to grow, and then we progressed into an NHP study. In the NHP study, you'll see on the right, we were able to demonstrate that same RNA replacement, at least 40% in our NHPs. And then also through durable expression, so 6 months in these NHPs, we were able to demonstrate it above 30% protein replacement, so ABCA4 protein replacement. We believe the marker for success in this in knockout mouse model is 10 to 20%, and we vastly exceeded that in NHPs with a safe and durable 6-month experiment. In terms of safety and tolerability, you'll see the OCT and ERG images are almost exactly the same from the pre-dose images through 6 months, so again, demonstrating safety and tolerability. And the image on the right shows the dose dependency of our exon editor. So as we increase the amount of exon editor applied, we see an increase in the RNA replacement. So we have safety, tolerability, dose dependency, boom, IND, we're off to the races. So our phase 1, phase 2 is a traditional dose escalation study with 3 different doses, a low, medium, and high dose. And after each dosing, we exchange with our safety monitoring committee, really with a primary focus of ensuring the safety and tolerability of our ABCA4 exon editor. A lot of our trial design, as well as some of the primary endpoints, is really fueled by an awesome natural history study. This allows us to set a baseline for not only the genetics, the known location of these mutations, as well as baseline what atrophy looks like in a diseased macula. We use 2 series of endpoints. So we use structural endpoints like FAF or OCT, and we combine that with a functional endpoint like microperimetry to be able to study the effect of our drug on these patients. We also use fellow eye normalization to really power our study to demonstrate the functional change of both that structural as well as functional changes in the macula. But our pipeline is much beyond just the eye. What you'll see here is that we have several, like I mentioned, several neurological targets that we've partnered with Roche, but our internal pipeline is really focused not only on neuromuscular, but as well as other targets. In each one of these targets, each one of these molecules, whether it's the clinical one or those in the discovery phase or research phase, these are single vector programs. So that hallmark Ascidian approach to exon editor applies to all of these. A single AAV vector, we're maintaining native gene expression. And we really believe what's differentiated for us is that we're knocking down the mutant form, and we're replacing it with the wild type form, all done in a single reaction. Our technology is only as powerful as the awesome people that we have at Ascidian. Listed here is our leadership team, but even more importantly, there are 34 other Ascidians that are in the Brighton area of Boston that are really fueling the research, development, and clinical. We have a fantastic and very empowering clinical and scientific advisors that support our organization as well as our board of directors. Lastly, in summary, I am super excited about the potential of RNA exon editing, not only for Stargardt's patients, but also for any other conditions where folks have thought that AAV is insufficient to deliver a functional therapeutic intervention. Exon editing, what I've shown here is it provides the durability of gene therapy, but without the need of a DNA edit, without the need of an exogenous or foreign enzyme. We can address traditional diseases that were untouchable by AAV due to the packaging limitations of AAV. And we do all this with maintaining the native gene expression levels. We can address both recessive and dominant genes. And again, we are eliminating or we're downregulating the expression of the toxic species while also restoring the wild type species. Our clinical program, ACDN01, we've demonstrated it both in vitro and in vivo exon editing, which really fueled us to open up the IND, and we're super excited to share the clinical data when available. Our partnership with Roche is further validation of our technology, and this is a lot of characterized by our very robust intellectual property portfolio. We have a broad pipeline, and more importantly, we're super excited to chat more about exon editing, whether it's here or whether it's in the hallways or at a breakout session. More than happy to share about the power of exon editing, and the future of Ascidian is bright. Thank you all.
Thank you, Ascidian Therapeutics.